And don't forget, if you want to look at your tests, I keep them up in the lab. You can look at anything you want in the lab. Which reminds me of this. I was actually looking at my wall this week, and this is kind of an interesting story, or I think it's interesting. This is part of my wall, yeah. My grandfather built this clock. My grandfather was the first, my great-grandfather was the first out of the Amish community. I look Amish, right? You can totally tell. He was the first out of the Amish community, and he used to love building things, but I really didn't get to know him very well. But there was one thing that I did learn from him, and this is kind of cool. One time I watched him going through a stack of wood, and he was throwing pieces of wood in one pile and the other pile, and he was throwing a lot of wood in this big pile, and this was basically his reject pile. This was scrap wood for bad stuff. But he was piling all this stuff, the good stuff, over on this side. And it was a really tiny pile, and all these pieces of wood, they weren't perfectly straight or anything, but he said, these are the pieces of wood that I use. And what was interesting is that when he said, why? He said, because when I go through the wood, I look at the type of tree it came from. It's not the type of wood, but the type of tree, where the tree was raised. And he said, this pile over here, I can tell that these came from down in the valley because they were protected. The trees were protected, they were nurtured by, the valley and the stream, and they didn't have to fight for anything. So their wood, if you look at the grain, it's wider grain, it's a softer wood, it doesn't hold up to abuse very well. He said, but these trees, they came from the top of the hill. They're exposed to the sun all day, they're beaten by the winds, they're abused by the storms when they come through, they're more likely to get hit by lightning, and they can feel the crash of the thunder. These have gone through a lot, and they're better for making furniture because they're stronger woods. They came from the same type of tree, but where it came from was a lot different. And I always think about this because I was sitting in my living room, it was 2 weeks ago now, on Friday, and I looked at the big empty wall, and I looked at this ugly wine rack my wife had, and I thought, that's got to go. And so I went to Menards, and I bought a stack of lumber, and as I was going through the lumber, this came back to me, and I'd totally forgotten about it. And so as I'm pulling through, I'm looking at the grain. They're all this lumber, I go for the crap wood. And I looked at the scrap wood lumber pile, and I started looking at the grain. It didn't matter if the boards were abused, if they had the right kind of grain, I started pulling them out. And when I take them home, I can trim them. I can make them exactly what I want because I know that wood's really strong. And this is actually what I built that weekend. So, yeah.
You built that whole thing?
No, I love building stuff. I'm not completely finished because you see one knob is missing right there, and I didn't get a chance to put the handles on these.
Can you make bunk beds?
But I have made bunk beds, yeah. But I can't make anything for Mercy students, sorry. But yeah, and this took me a weekend. I haven't stained it yet, but as I was building this, I love building stuff because I just sat there and I looked at the wall and I had a piece of paper and I just drew what I wanted and I went into the garage with the wood and started building it. But thanks. And
if I get out on Craigslist, then will you say I will make
maybe after you're done with Mercy.
Okay.
But yeah, it just kind of reminded me of that. It's the same situation. Some of the classes you take, you're like the lumber. Some of the classes you take, you're going to have to go through some adversity and you're going to hit challenges and you're going to hit things you're going, God, I just can't handle it. But what makes you different than a lot of people, or a lot of people when they do that, they just give up and they say, forget it, this isn't for me. Those are the people that are working at McDonald's, by the way. But it's the people that they endure it, they go through the tough stuff, they work their butt off. Even if they don't get the perfect grade, they still work really hard to get it and they understand the concepts. Those people that overcome adversity, those are like the pile of wood that they're not perfect pieces of wood, but what they're made out of is just incredible. So that's kind of like, when I look at the wood and I'm working with that, I start thinking about that. People are like the wood. The more adversity you go through, the more you can handle and the better you are to work with later. That took me the whole weekend to make, so 2 days. A what? Yeah. Right, yeah. Okay. Yeah, I have fun building that stuff. It's a blast. Just the right side what?
So you said a wine rack.
Oh, yeah, no, I started with wanting to build a wine rack, and I just went a little overboard. So I just framed the whole window and everything, and it was fun. Oh, yeah.
That's impressive. You should build a house.
I just want to go and compete with Pottery Barn because all the crap you can buy there, too expensive. You can build it at home for nothing. Okay, so cardiac muscle. Let's get going with this one. Lecture number 5. Don't forget that the homework's due, so make sure you turn the homework in today for number 4. What? The ion for calcium? Oh, how you write it out? Yeah, Ca++ or Ca2+. Yep.
I saw that in the book, and I was like, is that calcium still?
Yeah, I always write plus in my mind. I don't know why. It's technically Ca2+, but okay. So cardiac muscle, when you're looking at it, most of this, by the way, is an anatomy refresher, because if you don't get your anatomy with cardiac, you're going to struggle. So as we go through this, we'll talk about the heart, we'll talk about the blood, and we'll talk about the blood vessels, and they're kind of subdivided, and I split the homework into 3 parts too. So the first one's cardiac, and if you've already looked at that homework, it's primarily anatomy. I mean, and I'm not saying that because this is an anatomy class, but you have to know it. When we talk about the flow through the body, it's all about anatomy, where things go. All right, so the pathways. Like you should already know, what always flows into the heart? An artery or a vein? A vein. What carries oxygenated blood, the most oxygenated blood? I love asking that because it always takes me back to high school, and I remember my biology teacher saying, arteries always carry oxygenated blood. Is that true? No. The most oxygenated blood vessel in the body is what? The pulmonary vein. Yeah. So it's not always true. You have to know and understand your anatomy. And if you understand what's going on where the blood goes from the lungs to the heart through a vein, then you know that that's going to be well oxygenated. So go back and refresh. In fact, I'm making you go back and refresh your anatomy. Okay, so we'll talk about the blood. It's a transport medium. It carries all kinds of stuff. It carries nutrients, it carries oxygen, it carries CO2, it carries waste products, it carries proteins, it carries cells, it carries temperature, so it carries heat. It's a heat transport. You shake a muscle in one part when you're shivering, it heats up that blood there and then distributes to the rest of the body that can't shiver. The heart itself is it creates a pressure gradient. High pressure at the heart, which means that the pressure always moves from where? High to wherever it's lower, so it moves downstream. Why can't it flow backwards, though, if that's lower pressure? Because of valves. And we'll talk about the blood vessels. What are the only blood vessels that actually have those valves? The veins, yep. All right, so know your pathway. You've got the heart, you have the 4 chambers, you know the different types of circulation. What do you call the circulation that goes to all of the body except the lungs? Systemic. What do you call the part that goes to the lungs? The pulmonary, yep. So pulmonary circulation versus systemic circulation. So just knowing that, think physiology like. Which side of the heart pumps to the system? The left or the right? The left pumps to the system, so where's the right pumping to? The lungs. Which side has to have a higher pressure gradient? Right or left? The left. Why the left? Because it's pumping everywhere. It has to pump to the top of your head, to the tip of your toes. The right side only has to pump right next door to the lungs, right? So lower pressure. And these are concepts that are written in your notes, but I'm just asking you ahead of time to get you to think about it. You already know these. I know you already know these different things. If you know your anatomy, you can figure the physiology out really easy like. Right? So the pulmonary circulation, you already know, goes between the hearts to the lungs. Heart to the lungs. And back. And then systemic goes everywhere else. And we'll talk about the distribution of blood and how it goes to the different organs, where it's flowing. So the heart anatomy, you need to know these structures. The atrium, bottom or top of the heart? The top. The ventricles, the bottom. Yep. Which are larger? Ventricles. Which side is larger in the ventricles, the left or the right? Why does the left have to be... Well, wait. Which size muscular is larger? Let me get this. The left side muscularly. The volume of the left and the right, though, what do you know? They're the same. They're the same. You can't have more pumping on one side than the other. They both pump equally as they're pumping. When you look at the size, though, why does the left ventricle muscle size have to be bigger? Because it has to be stronger, right? It has to pump further. What kind of blood vessel always flows into an atrium? Always, regardless. If there's blood flowing into an atrium, it's from a what? From a vein. If there's blood, or what kind of blood vessels connect to a ventricle? An artery, because ventricles are always pumping where? Out, away from, yep, into the arteries. And then arteries and veins, remember, don't get mixed up and say arteries carry oxygenated and veins carry deoxygenated, because it's not true. Arteries always carry blood where? To or from the heart? From. Always pump away. Veins always pump into. Right. And then septums are dividers. And technically you have septums that go in between the atria, you have septums between the ventricles, you have septums between the atria and ventricles. So they're dividers between each of the sections. And we'll talk about them later. They're also there to help keep electricity isolated to different chambers too. So you should know your anatomy. What do you call these little strings? chordae tendineae, the heartstrings. Yep. And then these little muscles are called the papillary muscles. Yep. And then what's this thing here? You don't have to be specific, but in general, what's this called? It's a valve that prevents backflow. Yep. And when you look at the valves, you have these two valves. What are they generically called? They're between the atria and ventricle, so we call them the atrioventricular valves. How about these ones that are in the valves going out of the heart? Semilunars. Yep. And then the one that goes to the pulmonary circulation is called the pulmonary semilunar. And then the one that's going to the systemic circulation is called the aortic semilunar, right? And then you can see examples of septums. So here's a septum here, and technically you have septums here that divide the atria from the ventricles too. An artery, the blood always takes it away from, yeah, so arteries always take the blood away from the heart out of a ventricle. And then when you look at the size of the heart, the left and the right, you can see volume-wise, it's like one of those tricks where you get a short fat glass or a tall skinny one, which one holds more, and they trick you because they're both the same. When you look at the volume, they both hold the same amount. But when you look at the size of the muscle around it, there's a huge difference in the size of the muscle because the left side has to pump more pressure, higher pressure. So if I ask you a question like this, and again this kind of comes back to anatomy, which one of the following is false? Okay, yeah. So if you look straight at number 2, there are 2 atria and 1 ventricle. What's wrong with that? Yeah, there are 2 of each. And I don't remember other animal like biology that well, but I think that describes either a fish or a frog or something. But all right. So number 2 is the right answer. How about pathway of blood flow? You should know this. And again, this is anatomy, but when you look, you can pick any starting point you want as long as it comes back in a circle, which is why they call it the circulatory system. So you can pick any starting point you want. We're going to start with the superior and inferior vena cava. They flow into what? Yep, they flow into the right atrium. Do they flow through a valve? Nope, no valve there. And then from the atrium, the right atrium goes into the right ventricle. Is there a valve there? Yes. It's an AV because it's in between the atrium and ventricle. What valve is that? Do you remember? The tricuspid. Yep. And then from the right ventricle to the pulmonary trunk, is there a valve in between those two? Yes, there is. What's it called? The pulmonary semilunar. And then the pulmonary trunk, of course, you know the pathway. The pulmonary trunk, is it an artery or a vein? It's an artery. It's flowing out of the heart, so it's an artery. And an artery goes to what smaller structures? Do you remember? Arterioles. And then from arterioles down to the little capillaries, and then from the capillaries you come up to venules, and then venules turn into veins, and then you're coming through a pulmonary vein going back into what? The pulmonary vein comes back into the left atrium. Yep. So yeah, I kind of walk right through this, but the pulmonary trunk goes to the arterioles, down to the capillaries, and the capillaries are inside the lungs. And then from the lungs, you pick up oxygen, you bring it back through the venules, the veins, and into the left atrium. Is there a valve in between the pulmonary veins and the left atrium? Nope. Nope. And then from the left atrium down to the left ventricle, is there a valve in there? Yes. What's it called? Yep, the bicuspid. What's the other fancy name for it? The mitral, because it looks like the Pope's mitre. I hear they're getting a new Pope. That's exciting. All right. And then I thought a Pope had to die.
I did too. I didn't know they could have done it.
Yeah. This job sucks. I'm going to be a garbage man. Maybe. And then from left ventricle to aorta, are there any, yep, what's the name of the valve? The aortic semilunar valve. Yep. And then, of course, you know the pathway. From the aorta, it branches off, goes from aorta to the different arteries, and the arteries to the arterioles, arterioles to the capillaries and the tissues, systemic tissues, and then from the systemic tissues up to the venules, to the veins, back to the superior vena cava or inferior vena cava. Do you remember the difference between the veins in the head and the brain than the veins in the rest of the body?
Different colors. They do the opposite.
They don't have valves. In the brain, you don't have valves. Why would you not need valves in the brain?
Gravity.
Gravity. Yeah, you're using gravity. Right, so there's the pathway, and this is the picture out of your book that just walks you through. So you should know automatically the pathway. This is a really ugly, very full picture. But you can pick any starting point and go through and walk through that order. Right, so correct blood flow. Which one's correct? Look at your neighbor and have them help you through this.
Starts to right. Number 3. Trace.
What's wrong with number 1? So left atrium, left. Doesn't really matter though. It's a circle. You can start at any point you want though. So left atrium, left ventricle to the. What should it be then? Should be the aorta. So you know number 1's wrong. How about number 2? Right atrium, right ventricle to the aorta. That should be right atrium, right ventricle to the pulmonary trunk. So you know number 2's wrong. How about number 3? Right atrium, right ventricle, pulmonary trunk. True. Number 4, left atrium, right atrium, aorta. Yeah. So number 4 is wrong. Okay, right heart circulation is called the pulmonary circulation, of course, because it's pumping to what? To the lungs. Do you have to have high pressure? No, because there's very little resistance. You're going next door, so you don't have to push really hard to get from the heart over to the lungs. There's not a lot of tubing to run through. So it's low resistance, low pressure, and this pathway, low oxygen. That's the whole purpose for going to the lungs. What's it picking up? Oxygen. What's it dumping off? CO2. And then the left side of the heart, is it going to be oxygenated or deoxygenated? It's going to be well oxygenated. And is it going to have very far to go? Yes. When you pump out, you have to push to the brain, you have to push all the way down to the legs, all of the organs in between. So there's high resistance, which means you have to create high pressure to overcome that resistance. Are these already pre-written in your notes?
No.
Okay. So they're just exactly the opposite. With the right circulation, you have low oxygen content. Left, you have high. Right circulation, you have low resistance. On the left, you have high resistance. It's hard to push out of the heart. That's why the heart's so... What on the left side compared to the right? It's so muscular. Yeah, so big muscularly. And it creates a lot more pressure on the left side. And here's why. When you're looking over here, the right ventricle just pumps right next door to the lungs, and this is actually seems like an exaggeration for the distance that it has to go to the lungs, and then pumps right back in. But the left ventricle has to pump to all the muscles of the body, to the kidneys, to the GI tract, to the brain, to all the other organs, all the way down to the tip of your toes. So lots of pressure. As it's pushing this pressure is even across here. What's interesting is that each of these organs decides how much blood flow they get in. So of all these organs, the brain, the kidneys, and the muscles all require roughly 20% each. Seems weird because a lot of your muscle or your mass is muscle. Your kidneys, little tiny things are like 1% of your body mass, and the brain's less than 3 pounds, so still less than 1% of your body mass. But they still require about the same amount of blood flow as muscles. It's amazing. All right, and then the valves. Valves prevent backflow. And you already know the names, you already know where they go. The only other structures that we haven't talked about yet were the chordae tendineae and the papillary muscles. So the chordae tendineae, they attach to which valves? The semilunars or the AV valve? AV. And it helps you anatomy-wise to distinguish between the two types of valves. Why would the AV valves have to have these special support cables compared to, well, anywhere else? What are they doing? They're preventing all that extra pressure from moving backwards. Man, when you squeeze really hard down here, you're going to squeeze up and out, but if you didn't have these cables, what would happen? You'd blast it backwards, right? You'd have what they call prolapse. It would pop backwards and the blood would flow backwards. When you look down, you can see the direction of the blood flow. You have that nice smooth curvature, so it's kind of like this. When my fingers are like this, they can't push backwards, but they can push in. So it's easy to push something this way, and then when they snap back, they catch, and I can't overextend them. So when you're looking at the valves down through the top of the heart, that's what you see. And then there are the chordae tendineae and the papillary muscles too. Okay. So when you're looking at the valve open, you can see this nice smooth flow, and then when it comes back, it kind of loops underneath, catches the valve, and snaps it shut. And we'll talk about these types of flow later too. So which one of the following is true about the valves of the heart? How about number 1? The AV valves are between the arteries and the ventricles. What's wrong with that? That's A and V, right? It's between the atrium and the ventricle, right. Make sure you read the question slowly. So number 1's wrong. Number 2, the AV valves open down into the ventricles. Sure do, yep. They open down to allow blood to flow down and into the ventricle but prevent it from flowing backwards. So that one's true. Let's double check on 3 and 4. Semilunar valves have chordae tendineae? Nope, the only valves that have chordae tendineae are the AV valves. How about another name for the left AV valve is the tricuspid? What's the other name for the left AV? The mitral or the bicuspid? Right, when you look at the structure of the heart actually has its own type of skeleton. It's not made out of bone, it's made out of fibers to help hold it in place. In fact, if you looked at the muscles are designed really cool, so they weave and they kind of turn into like this circular pattern, so when the heart contracts, it squeezes and rings. So it actually does this little mild twist, like when you're wringing a sponge to get all the water out. The heart does the same thing. It twists with that fibrous skeleton and squeezes. The other part of the fibrous skeleton is to help suspend the valves in place. So if the valves were just floating there and just dangling, they could collapse on themselves. The fibrous skeleton also gives support, so it holds the valves all in that nice smooth plane across there. Okay. When you look at the layers of the heart wall, you have to remember the layers. What's endo mean? Inside, it's within. And then myo means muscle, and then epi means superficial or on the outside. So when you're looking at the different layers, the endocardium is this layer down here on the inside. It's like Teflon. How many people here don't know what Teflon is? I don't even know if you can buy pans with Teflon in them anymore, but Teflon's this non-stick coating in pans, right? This endocardium is like a non-stick coating. Why would you want it to be non-stick? What's touching it? What kind of fluid? Blood that likes to clot, right? If it were sticky, what would happen? You'd clot all over it. It has to be smooth. It's real. When you've taken the heart apart, you probably noticed it's real shiny looking. It's real smooth. It's so that blood can flow through and it doesn't stick. If there are any flaws in this endocardium, any little flaws at all, those little platelets would start clumping up there and causing a scab, but technically it's a clot, right? So it would start scabbing up. It's the same problem that happens when you cut a blood vessel. When you cut that smooth Teflon layer, the clot recognizes that Teflon's broken, and the platelets, and they start moving over there and they form a clot. So this endocardium is a smooth lining, and it's continuous with the blood vessels. There's no break. So that as you're pushing the blood up and out, it's a nice smooth slip and slide pathway. It goes right up and out of the heart. One of the flaws when they make artificial hearts is that it's really hard to imitate this and this super non-stick lining without any breaks and any flaws going continuous into the blood vessels. So the major flaws that they've had to determine were, how do we fix that slippery surface so that there are no breaks at all? Because otherwise you're going from an inorganic structure, this fake heart, to organic tissue. There's got to be a nice smooth transition, otherwise it clots right where you transition.
Then how does it pulsate?
Because that's organic tissue. Yeah, with pig valves, they're real living tissue. They can actually merge right in. Myocardium is a muscle layer, and you look how thick it is. What's interesting is that all of this blood that's inside the heart, can it seep into the muscle? No, because of the endocardium, the slippery non-stick substance. The blood slides through, so all that blood inside your heart is actually not getting into the muscle of the heart. So the muscle of the heart has to have its own pathway to get blood flow. Anybody remember what that's called? The coronary circulation. Yep. So the myocardium is the thick, dense stuff that does the contractile. What type of muscle is that? Thank you, cardiac. The silence scared the hell out of me, I'm just telling you. What type of muscle? Skeletal, smooth, cardiac. We're going to go with that cardiac. Yeah. And then the epicardium is a superficial layer that protects the outside of the heart. It forms a sac, which we're going to talk about in a couple seconds or a couple slides. So you have this protective sac that goes all the way around the heart. Just like you have protective covering around bones, the periosteum, you have this peri-structure that goes around, and we'll talk about that. But we call it the epicardium. Okay, and then you can see the layers again. You've got that smooth stuff in here, that little trabeculae. It looks like Freddy Krueger's face to me. It's so cratery, but it's smooth. And then you have that thick muscle in here, and then you have the epicardium that surrounds the outside, which we'll talk about in just a second. We'll call it the pericardial sac, actually. All right, intercalated discs. When you're looking at this cardiac muscle, one of the unique features that makes cardiac different from both of the other types of muscle is this thing called an intercalated disc. So when you look at it, here's a muscle cell, cardiac muscle, just in case you forgot, and then you have this tight, thick, strong junction. I wish I hadn't used the word tight because there are tight junctions that are like Velcro, and there are these desmosomes that are like concrete and steel wire. When they poured the driveway at my house, what was interesting is that I watched them do it. Actually, I don't know why it was interesting watching them, because when I was a kid, my family were construction workers and they did this. But why do they put that steel cable through the concrete? Because if you take concrete's like chalk. If you stand on a piece of chalk, what's it do? It'll shatter, right? So concrete can only handle so much weight before it starts actually crushing itself and shattering like chalk. What they do with the steel bar going through there is it holds it together more tightly. So they put the steel bar through. Your heart has the same concept. It runs this fibrous material, this collagen-like material that's like a steel cable, between two heart cells, and then it dumps this plaque, this concrete-like plaque over the top, so it secures it. So that when your heart's pulling like this, what's it not going to do? It's not going to rip apart, right? If I used a tight junction, that waterproofing like Velcro, what's Velcro do when you pull it? It unzips, right? So this desmosome, really strong junction, holds the heart muscle together. This is one of the special features of an intercalated disc. Strong support, so they don't rip apart. That would be bad. And the second feature are these little things called gap junctions. So you have these little tube-like structures so that when this one gets excited, what's the next one next to it do? Gets excited with it. Yep. These are the two main features of an intercalated disc. This is a unique feature to cardiac muscle. Do you have gap junctions in skeletal or smooth muscle? And smooth, yeah. What kind of smooth? Multi or single unit? Single unit. But you're not looking at these super strong desmosomes. In fact, you can see these when you look at cardiac muscle. You can see intercalated discs are really dark structures in between the muscle cells. What do you think this pink stuff is? Myosin. What do you think the blue stuff is? Actin. Yep. So look at the arrangement or the organization. What's it look more like? Skeletal or smooth? Skeletal again. Yep, which gives us that striated appearance. Okay, so intercalated discs, you want to remember the desmosomes and the gap junctions in the intercalated discs. They're for strength and so that they can have autorhythmicity. They talk to each other. So do they fire as a multi-unit or a single unit? As a single unit. And a syncytium, the best way to think of a syncytium is a group of cells that talk to each other at one time. So when we talk about the atrial syncytium, we're talking about a pathway of cells that are all connected by gap junctions across the atria. So that when you fire part of the atria, what happens to all of the atria? It all fires in sync. Yep, so a syncytium is this connective pathway. It's called a syncytium. So connective pathway connected by gap junctions so that when one is activated, they're all activated in unison. So it helps them fire like a what? A multi or a single unit? A single unit. That's... No, because syncope is actually feigning. That's a totally different concept. But I like how you're looking at the words because I always do that with words too. It gives you meaning. Okay, pericardial sac. So the pericardial sac, what layer is this made out of? The endo, the myo, or epicardium? The epicardium. So when we talk about the pericardium or the pericardial sac, it's the epicardium. It's out on the outer surface. This sac has two special layers and then a cavity on the inside we're going to talk about. So first you have the layer that touches the actual organ. When we refer to the internal organ, what's the V word that refers to the organs? The viscera, right? So the visceral layer of the pericardium is the one that does what? Or touches the opposite side or actually touches the organ? It actually touches the organ itself. The opposite side of the sac that's away from it is the parietal. And here we go. So if you look at these layers, you can see there's no break. It's a continuous layer of the epicardium all the way out here, and then it just folds over itself and comes all the way back around, up and around, and then back in again. There's no break. It's like a water balloon. If you took a heart in a water balloon and you took the heart and you push it down the water balloon would start wrapping around the heart. It's the same idea. You didn't break the balloon, it just wrapped around it. What benefit would you have to having this water balloon around the heart? a little bit of shock absorber, a little protection. I heard the L word. I thought I heard it. It helps lubricate to reduce friction. Yep. Think about it. Your heart, it's tissue, right? Just like your hand and your palm. If you took your heart and you kept going in your chest all day where it was rubbing tissue on tissue, what would it be doing? Imagine, creating heat for one thing, friction. And then what would happen to your knuckles if you did that 24 hours a day? Yep, you'd start wearing the surface down. So it's a lubricating area, and it reduces friction. So if I look at the layers again here, I have my endo. What's this layer of muscle called? The myo, and then you have the epicardium here, and this part's called the visceral. As it wraps around, it causes the parietal. And then technically, if you go into cardiology, there's an outer layer called the fibrous layer that helps hold everything or secure everything in place. But you need to know the visceral, the parietal, and then the cavity in between is called the pericardial cavity, which literally means the cavity around the heart. And then the fluid inside is called pericardial fluid. Not too tricky. Okay. Now we're going to start, this is kind of like, you might want to put a line above this because we're going to start talking about the electrical activity of the heart now. So those were a lot of anatomy terms that you've gone through. Now we're going to start talking more physiology. And the first of the two types of cardiac cells, you have contractile and you have autorhythmic. The contractile cells, their whole purpose is contraction. They're workers. They're dumb. They just stand there and they wait to be told what to do. So when they tell them to contract, they go contract, and they contract really powerfully. The autorhythmic cells are like the foreman. They tell the contractile cells what to do. These are the ones that they send an electrical potential from one to the next to the next to send a signal that says, tell your contractile cells to fire now. These autorhythmic cells, they have this pacemaker activity. Does your nervous system have to control them? Nope. It can help modulate them or modify them, but they work on their own. And that activity that fires at a regular rhythm, we call it a pacemaker potential. If you look at the amount of cells, the contractile versus the autorhythmic, you have about 99% of the heart cells are contractile. Only about 1% are autorhythmic cells. The autorhythmic cells act like neurons where they're a real thin pathway and they tell the contractile cells what to do. The contractile cells are the bulky workers. Right? And here's an example of a pacemaker potential. Look what you're missing on a pacemaker potential. I pointed out earlier, did anybody remember what I said was gone? There's no resting. They don't rest. They go up, they fire, they start repolarizing, but when they get down where it would normally be a resting potential, they start depolarizing again. There's no stopping point. So as you're firing this, you've got depolarization, threshold, thump. So you have this excited potential shooting across that cell. It goes up here, it's depolarizing. At the peak, it starts repolarizing. You go back down under threshold, it starts resetting, but it doesn't go to resting, and then it starts depolarizing again. So you've got this slow depolarization, and then threshold, thump, an action potential, or technically a pacemaker potential. What you need to notice or figure out is look at what's causing depolarization here. What caused depolarization in a neuron? Increase in sodium coming in. What's causing the depolarization in this one? Calcium. Calcium leaking in. So calcium's leaking into the cell. As calcium leaks in, if it were a contractile cell, what would it start doing? As calcium came in, what does calcium make a cell want to do? A muscle cell want to do? It makes it want to contract. This is the autorhythmic, though. This is not a contractile cell. What's it going to do? It's going to make it depolarize. It hits threshold and it gets this electrical potential. So it sends a signal all the way through its syncytium and tells them all, fire. And then at the very peak, guess what happens to all those calcium channels? They close. Then what happens? Potassium opens. What opened on this pathway in a neuron? Potassium. You might want to watch for themes. Okay, so the big change here was instead of sodium going into a cell, it's what? Calcium. Calcium's depolarizing the autorhythmic or the electrical cells in the heart. Potassium repolarized neurons, it repolarized the muscle cells, it repolarizes these electrical cells. Potassium is like the repolarizer in all the cells we're going to talk about. So it's kind of a theme. Potassium's the repolarizer. And then as the potassium leaks out, it comes down here at the bottom, it's starting to reset everything, it's starting to close the potassium doors, those calcium doors are starting to open again, the calcium's leaking back in slowly, and then threshold, poof, all the calcium doors open. Calcium floods in, the calcium doors shut. So it's the same kind of pathway, up and down, over and over again. And it doesn't hyperpolarize. Do you want your heart to hyperpolarize?
No, but it's potassium, so no.
What would happen if your heart hyperpolarized? Huh?
It's still potassium, though. No potassium going to shut slowly. So
They do shut slowly, but the calcium's opening at the same time. So as potassium's still leaking in, calcium starts leaking in. So it's kind of like they're just exchanging places. And the calcium, look at the calcium compared to the potassium. What were you going to say? There you go. That's the way to think of it too. As one potassium moves out and one calcium moves in, which one's carrying a bigger charge? Yeah, so it's a little bit more powerful. Right now, for the sake of what you need to know, don't even pay attention to it. Yep, so don't even worry about the sodium right now. That was a good question, but yeah. Strike that from the record for now.
What were you going to say if the heart hyperpolarized?
Oh, yeah, what would happen if the heart hyperpolarized? If you hyperpolarize a neuron, that means you can't fire it. What would happen if you hyperpolarized your heart? You're screwed, yeah. That's the last thing you ever do. Yeah, so you want to avoid hyperpolarization in the heart. And we'll talk about that more when we talk about the contractile cells too. Okay, autorhythmic cells, when you look at them, you need to look at the pathway of the autorhythmic cells. And I'm just going to flip to the picture. It all starts here in a normal, healthy heart. And I'm going to keep saying that normal, healthy heart over and over and over again. Because if you break this heart in any way, you screw up this pathway. So it should all start with the sinoatrial node firing first. There are pacemaker cells all over your heart. You have some here, you have some here, you have them down here. But the key is that the fastest ones are the kings. Because if this one fires, what's going to do to all of the other ones? Makes them all fire at the same time, right? So they all reset at the same time, and if this one fires again rapidly before this one can depolarize, what's going to happen? It fires them all again. If this one shuts off, who's going to take over? This, right, you know a little bit more than I want you to know, but you're exactly right. The AV would take over. But do you know why? Because it is the second fastest. Yep. So if the fastest one is lost, the second fastest one takes over. If the second fastest one is lost, what takes over then? The third fastest, exactly. So it goes in that order. I love it. You know, you look at the heart and you think, wow, this is amazing how this thing works. It's not a really smart device. It's really simple. If the fastest one stops firing, the second fastest one takes over. If the second fastest one stops firing, the third fastest takes over. It's just like a chain reaction. If you break one, then the next leader would take over. All right, so it all starts here in the right atrium up at the SA node, sinoatrial node. So if you have a sinus rhythm, it means that your SA node's firing appropriately. It's leading the pack and it's doing what it's supposed to be doing. Exactly. If those two stop working, then you actually depend on the Purkinje's down here, or the bundle of His. The slower ones down here will fire, and they'll send a signal, but they send it backwards. This isn't like a neuron where it's a one-way pathway. If this pathway fires here, it can actually fire backwards to those gap junctions, and will fire in reverse. Good? Huh? That would suck, right? Because what do you know about the heart? The atria have to fire, then the ventricles, right? Because the atria have to push the blood down into the ventricles, then the ventricles fire. In this situation, the ventricles are firing, and then the atria are trying to fire after it, or at the same time. So think about if they try and fire at the same time. Which is going to win? Little dinky atria or this huge, powerful ventricle? The ventricle. Yep, and we'll talk about those malfunctions later too. So the pathway starts with the SA node, and then you have the syncytium that shoots across. So that all of the atria will fire at one time. You got the right atria firing at the same time as the left atria. You've got this electrical potential that shoots from one gap to the next gap to the next gap to the next gap, all the way across, so that all of the atria go and they push down simultaneously. So that when it pumps, the left and the right sides pump at the same time and push down. This is kind of cool. I like how this works. The AV node is a delay node. It takes that electrical signal that shoots down here, and it kind of holds the signal. It catches the power for a second. It holds the electricity so that what can happen completely? So that you get flow. Exactly. So the atria get a chance to contract and squeeze the blood out before the electricity hits the ventricle. So with the AV, you want to remember there's an AV delay. The AV holds onto it for a few milliseconds, holding onto that electricity so you get a chance to flow the blood down from the atria to the ventricle. And then, once the atria is done contracting, then the AV node lets go. It lets go of the electricity, and it's going to send it down to this thing called the bundle of His. It's not her bundle, it's His, right? And then it splits. So here you have the interventricular septum. It means it's where? Inter means between, and ventricle is right. So it's between the ventricle septum. So this electrical pathway goes down through the interventricular septum. It branches, so they call it the left and the right bundle branches. So it goes from the bundle of His to the left and right bundle branches. And then it goes down to the apex of the heart, the very tip. At the apex, then it turns into, from the bundle branches, into these things called Purkinje fibers. And the Purkinje fibers, you have to imagine the heart as being three-dimensional. I know this is a flat picture. But once this goes right down the center of the heart, when it gets to the tip of the heart, now it's branching out in 3 dimensions. Like each of my fingers are Purkinje fibers. They go up around the outside of the heart so that it stimulates the whole outside edge of the heart at one time. Whoops, wrong way. So it stimulates here, but remember in 3 dimensions. So it starts squeezing at the apex, and it starts squeezing from here up. Why would it start squeezing from here up? What would be the benefit? It pushes the blood up, so it pushes it up and out of the top of the heart. So as the apex starts contracting, all these cells, like dominoes, they all fire together. It pushes up. And that's the electrical pathway. If you screw up this pathway, like for instance, if you cut the bundle of His, is the right atrium okay? I mean, is the SA node okay? Yeah. Are the Purkinje fibers okay? They're alive, right? What did I tell you about their pacemaker ability? They all have pacemaker ability. So if you lose this bundle of His, what's actually happening? Are the pacemakers still firing? Yes. Are the SA nodes still firing? Yes, but are they talking to each other anymore? No. Your heart starts acting like 2 separate hearts. The atria fire at one rate, and the ventricles fire at their own rate. So they're out of sync, and we'll talk about that too. So that's why you want to know this pathway. Everything has to be in sync for your heart to contract and move appropriately. And then there's the list. All right, and then here's your autorhythmicity again. So when you look at this repolarization and depolarization, here, what was going into the autorhythmic cells? Calcium. Calcium flowing in slow here, you hit threshold and all of the calcium doors open. What's calcium going to do now? Rush in really, really, really quick, right? So it's flooding in at the very peak. What do you do to those calcium doors? You start shutting them, right? What are you opening? The potassium doorways. What's potassium doing? Rushing in or out? It's rushing out, causing repolarization. And then at the very end, then the calcium doors start going, shutting. But what's happening to the potassium doors are shutting. What happens to the calcium doors? They reopen again. So now you're starting to get calcium flowing back in, never giving you that resting potential. It's constantly in a flux of depolarization, repolarization, depolarization, repolarization. Back and forth and back and forth. What would happen if I opened more calcium channels sooner? It would go faster. I would depolarize faster. What would happen to my heart rate? This would go quicker together. What would happen if I gave you a calcium channel blocker that closed some of those calcium doors and prevented them from opening? It would slow the movement of calcium, causing the heart rate to go slower. So you can see the difference. Here's with more doors open, a faster rate. Block some of those doors and it's slower leaking in. It's like an hourglass. When you hit that threshold, enough of the calcium leaks in, then boom, it looks exactly the same up here, whether you're fast or slow heart rate. The only difference is that rate going up into depolarization. And it's all because of what ion? Calcium. So what do you know is true about the autorhythmic cells? Yeah, what's wrong with number 1?
It's 1%.
Right, 1% are autorhythmic, 99% are contractile. So you can cross out that one. How about number 2? Autorhythmic cells make their own electrical impulse. That's actually true. Yeah, let's make sure we go through the other ones though. Number 3, depolarization in an autorhythmic cell is due to sodium entering the cell. It's calcium. So you can cross out to number 3. How about autorhythmic cells have a resting membrane potential of about -50? They don't have a resting potential. Right. So number 2 had to be our right answer. They're autorhythmic. They make their own electricity, their own rhythm. Yep, using calcium going in and potassium going out. Okay, autorhythmic activity, the pacemaker. What should be your normal pacemaker? What do we call the pacemaker of the heart? The SA node. Why is the SA node the pacemaker? It's not because it's so smart. It's the fastest, exactly. It has the fastest depolarization rate. The latent pacemaker, you want to think if the SA node's broken, what's next in chain of command? I heard Jerry say it earlier. The AV node. Latent means it's delayed. Yep, so what's going to happen to your heart rate? It's going to be slightly slower. The term complete heart block means that you lost the AV node. Your AV node's broken. So this is out of the picture completely. Is the SA node still working? Yes. Are the Purkinje fibers still working? Yes. What happened? They don't communicate. A third-degree heart block is when your heart's looking like it's two different hearts. You basically have this double rate. You have the rate of the atria and you have the rate of the ventricle, but they're not the same. And when we look at lab this week, we're doing ECGs, and you'll see, we can look at an electrical rhythm, and you'll see that the electrical rhythm of the ventricle is actually not the same as the electrical rhythm of the atria.
How long did that go on for?
Not a long time, but it can actually go on for a while. Yeah, and we'll talk about it in lab too. Depends on you. Depends on how much activity.
Like how long
Yep.
does it lie?
It's a good question. It's a great question. It depends on the person. I had this asked in last week in one of my, actually I was subbing, it wasn't even my class. In pathophysiology, someone was asking about how fast you break down alcohol, and it actually depends on the person. It's the same idea. Your genetics have a lot of factor. It depends on your activity. Like people that they're inside, they're making a sandwich, they're fine, they're watching TV, they're fine, they go outside, shovel the snow, and what happens? They have a heart attack. I mean, it's just the demand of the activity. They would have never had that heart attack had they wouldn't have had the heart attack then if they hadn't gone out to shovel the snow. So it just depends. Some people, with a very sedentary lifestyle, they may be able to make it for a while, but you'll start seeing like blood flow problems. They might have clotting issues, which could, 2 seconds after having a third-degree heart block, you may throw a clot and boom, you're dead. But somebody else may have that third-degree heart block for weeks or months without it being detected. Yeah. Yep. It's all about what's happening besides just the actual heart block. Huh? That could be a situation, and that kid probably or most likely had a genetic. If it's a kid how old was he? Do you know? High school. If it's a genetic disorder, or if it's a kid that dies of something like a massive heart attack, usually it's genetic. It's called Prince-Metal syndrome, where they could die in their sleep. I mean, a kid goes to bed and then they die in their sleep, and they're like, "What the heck happened?" They had a heart attack in their sleep. All right, so complete heart block. We also call it a third-degree heart block, which I'll talk about first and second-degree heart blocks later. But actually, a third-degree heart block is also known as complete heart block, and it's because the AV node's gone. It could be the bundle of His though too. But typically it's the AV node. It's making the heart work like two different hearts. Ventricles and atria are not talking to each other. So an artificial pacemaker, what they do is they actually monitor the heart. They're a little device that monitors the heart, and if it misses a beat, it zaps it real quick, and it controls that extra heartbeat. So an artificial pacemaker is basically anything that's helping pace the heart. This is an interesting one. An ectopic focus means an abnormal location for the pacemaker. An ectopic focus is referring to an abnormal location. Focus is talking about location. Ectopic means abnormal. That's stimulating the heart. If you reach in and you start stimulating some of these Purkinje fibers, if you rub them, it'll make them start firing. So if you rub them at a fast rate, what's it going to make them start doing? Firing fast. What could that do?
Stimulate your heart.
If the pacemaker is now firing faster, the Purkinje is now firing faster than the SA node, what's going to happen? The signal goes backwards. Yeah, bad news. So any type of ectopic focus, if you have something that's rubbing the heart abnormally in a weird way, like if you have that pericardial sac becomes inflamed and now it's creating friction on the Purkinje fibers, what could happen to their heart rate? It starts increasing, right? It could actually misfire. Let's say they rub it wrong this time and it creates a heartbeat really quick, and then they wait, and then they misfire a couple times in a row. What kind of heart rate would you call that? Arrhythmic, yeah, so an arrhythmic. Ectopic focus, what you want to remember is an abnormal location to start the heartbeats. So sometimes, and I'll show you a picture with the Purkinje fibers misfiring, but that would be an example of an ectopic focus. Not supposed to happen in a normal heart.
When
Yeah.
doctors are in surgery, they can't get the heart it to start and they start manually pumping it?
That's what they're trying to do. They're trying to do the same thing. So, and if they can't start it on their own, the pumping is just trying to get the blood flowing so that you don't have a clot that builds up in that one spot. But normally, what would they do? They wouldn't just rub it to start it. They'd shock it, yeah, because that's way more effective. Okay, here's an example, and I love this analogy. This is the analogy your book uses. So they compare the heart to trains. When you look at trains don't always just have one power car. Sometimes they have at one end and they have another one at the other end. It just depends on where you're trying to go. But what if you had these 3 power cars? I don't even know what you call those with the engine in them. But if you had 3 of them connected, they're hitched together. All right? If the fastest one's going 70 miles per hour, but the other ones are connected, how fast is the train going? It's going 70. What would happen if you uncoupled this train car? Yep, that one would go at its own rate, but the others would all go at 50, right? So if you lost this one, if you knocked it off the track completely, the whole train is going 50. What would happen if I knocked this one off? How fast is the heart going? It's going 30. What would happen if I just knocked the AV node out completely, but the SA node and the Purkinje's were still on the same track? The SA node goes 70, and the Purkinje's go 30, exactly. What kind of problem was that?
The two fire at different rates.
They fire at different rates, which is called a complete heart block. Yeah, so that's an example of a complete heart block. The SA node's going at one rate, the Purkinje's at another rate. They're not working together. What if I had an ectopic foci and I started rubbing, or I had some friction that was stimulating the Purkinje fibers and they started going like 120 miles an hour? How fast is my train going? 120. Yeah, so my whole train is going at the speed of that ectopic focus, that abnormal location stimulating it. This one train is pushing the other ones, right? They're going, oh, they're along for the ride. I animate things too much in my mind, but anyway, there's your derailing, which is an example of a complete heart block, and then there's your ectopic focus, where the whole thing's going too fast. And then 3 criteria have to be satisfied for efficient spread of excitation. So getting that proper order is important. SA to the AV to the what? The bundle of His, and then to the bundle branches, and then to the Purkinje's. That's the normal order of things. All right, so the first thing, 3 things that have to be satisfied. First, atrial excitation and contraction should be complete before the ventricular contraction. In other words, what structure is preventing the excitation from getting down to the Purkinje's to stimulate the ventricle? The AV. The AV node has to create a delay, in other words. So the atrial excitation and contraction has to be complete, and it's because that AV node delays. And technically it's about 100 millisecond delay, so a few milliseconds. Few is a funny word. So it creates that slight delay so you can get the atria going and squeezing, contracting. Next, excitation of the cardiac muscle fibers should be coordinated to ensure that each heart chamber contracts as a unit to accomplish efficient pumping. Part of your atria can't contract by itself. The whole atria has to contract together. If you have this balloon and you just, instead of squeezing the whole balloon, you just push on one edge, does that make much of a difference? No. You want the whole thing contracting or squeezing at one time. The atria have to contract. If part of the atria contracts and the other part relaxes, what actually happens to that fluid? Nothing. It doesn't move. It just shifts around the atria, right? If blood's not moving efficiently, what are you risking happening? If blood's not moving, it must be clotting, right? It's clumping. If the blood sits stagnant in any area of your flow for too long, you risk a clot, right? That's why when you give somebody a tourniquet and you keep it on for too long, what are they worried about when you take the tourniquet off? A clot moving up, right? So they're afraid of that clot moving upstream. And then the third factor, so remember, the whole atria has to contract, the whole ventricle has to contract in unison. So just one little part doesn't do it. It has to be the whole atria or the whole ventricle at one time. And then the last, the pair of atria and the pair of ventricles should be coordinated so that they fire simultaneously. So the pair of atria have to fire together, and the pair of ventricles have to fire together, but do you want the atria and the ventricles to fire together at the same time? No. So they have to be coordinated so the upper pair fire, then the lower pair fire, and then the upper and then the lower. They have to switch off and on. So 3 criteria that have to be met. And then the spread of the impulse, you already know this, but it originates in the SA node, goes to the AV, to the bundle branches, and then eventually the Purkinje. Okay? So I already talked about this enough time. I don't think we need to talk about it anymore. And I already talked about this too. So it goes through the ventricle muscle from the epicardial surface, so it goes up to the outer part, and then it goes down and in. So it starts at the epicardium, the superficial part, and then the Purkinje fibers take it down deep into the myocardium. And there's your pathway. So it goes across here. Remember, you have that septum that divides the atria from the ventricles so that the electricity doesn't go. The only way that electricity can get from the atria to the ventricles is through what structure? The AV node. It has to go through the AV node. Then it goes to the bundle hist and the bundle branches and the Purkinjes. All right, AV node. The delay about 100 milliseconds, so about 1/10 of a second. And then the whole purpose, you already know this because I've said it 3 times at least, so it must be important. It's there so that the atria can depolarize and contract completely before the ventricles have a chance to depolarize and contract. Put a line under that because I don't want you mistaking this slide with what I've talked about before. The resting membrane potential of a contractile cell is negative 90 millivolts. Why do I not want you mixing this up with the last section we just talked about? The last section was about autorhythmic cells. It was about electrical potentials. This part is contractile cells. They're different cells. Contractile cells rest. The resting potential is about negative 90 millivolts. So the key here is this cardiac contractile muscles, not autorhythmic. When you look at their potential, does this look like an autorhythmic potential? Yep, this looks nothing like it. This looks more like an action potential because it has a resting phase, but look at that weird shape. It's crazy. So here, in this situation, this looks more like an action potential because look at what's depolarizing. Sodium again. What depolarized an autorhythmic cell? Calcium. So when you look at the contractile cells, it looks more like a neuron because it has a resting potential and it has what depolarizing it? Sodium. Look how fast this depolarizes though. Remember when we looked at the neuron, it was like slow, slow, fire. And then it was still kind of slow up at an angle. This is rapid. All of these doors, super fast. They're extremely, extremely fast opening doors. So as soon as I stimulate this contractile cell, all the doors open, you get a huge flood of sodium in. At the very peak, look what happens. Boom. All of the sodium doors slam shut. What door is going to start opening now? What do you normally want to think opens? Potassium. Potassium's going to creak open just a little bit, but look what's going to flow in at the same time as potassium going out. Calcium. What's more powerful? Calcium. So this is the unique feature that you're going to see that you haven't seen before. It's called a plateau phase. You get this rapid depolarization, and as it starts to repolarize, it actually levels out for a little bit. So it doesn't get a chance to repolarize completely. It plateaus during that phase. And it's because of the calcium. And calcium's slowly leaking in as potassium's kind of slowly leaking out. And what's happening? As the calcium leaks in, the potassium leaks out, and it's staying level. So you've got this weird leak. And then right here, suddenly all the calcium doors shut, and what's going to happen? Potassium's still flowing, but now potassium is becoming more dominant, flowing out, causing repolarization. What repolarized an autorhythmic cell? Potassium. What repolarized a neuron? Potassium. So it's straight across the board. This plateau phase because of calcium that characterizes that cardiac contractile cell. So what do you know is true about the contractile cell action potential? How about number 1? Repolarization in the contractile cell is due to calcium entering. No. What's repolarization caused by? Potassium leaving. So, and by the way, does calcium want to enter to repolarize? If calcium enters, what's it trying to do? It's trying to depolarize it, but it's coming in trying to depolarize at the same time that potassium's flowing out trying to repolarize, so you level. You know number 1's totally wrong. Number 2, the contractile cell has a plateau phase which is due to sodium entering the cell. What does sodium do to the contractile cell? It depolarizes it, so you know number 2 is wrong. How about number 3, depolarization in a contractile cell is due to sodium entering? Yes. What causes the plateau phase? The calcium. Yep, and that's it. So 2:30, yep, make sure you take online lecture quiz number 4 before we come back in. It's due by Thursday. The online lecture quiz is due by Thursday. You should take a peek and try and get that first homework for the cardiac section done. Like I said, it's a lot of anatomy, So you should already know most of it. And then the rest of it we've talked pretty much about already. Huh? I'd like to have it turned in on Thursday, but I'll wait until Tuesday if you have to. But
What, homework 5?
yep, homework number 5. Yep, because we're gonna homework 5, 6, and 7 are all going to be due before 2 Tuesdays from now. So not this Thursday, not this coming Tuesday, but the following Tuesday.
Jesus.